So, hello friends, in continuation with the RNA extraction method. In previous lecture we have learned that the different type of samples which we can utilize for the extraction of RNA and different type of methods which can be employed for the extraction of RNA, that is either kit based, either manual, or either by using both the methods. And where I have explained to you that the what are the precautions or general information you should have in your mind before isolating your or before using the kit based extraction of RNA. So as I've explained you that the whole blood has RBCs plus leukocytes, and RBCs are 1,000 times more than leukocyte. So if they are in huge number, but we don't need this group of population, these group of cells, and we need just leukocytes to be used for final extraction of RNA, because RBCs have very less or negligible amount of mRNA, one thing. Second, the heme part of the hemoglobin, it will inhibit the further downstream applications of PCRs and all that. So whenever you are using whole blood, you need to ensure the complete removal of RBCs from your population. And then once the RBCs are removed, then you can use these leukocytes for the RNA extraction. The other method, a popular method, is students, they go with the PBMC isolations by 5-col graded density gradient method, and from the PBMCs they isolate the RNA. That is also okay to use that, but if the students they want to use whole blood, it is also possible. So there are several companies, they are providing different kits for the extraction of RNA. The basic protocol or the basis is same in all the kits, most of the kits, and here I am explaining QIAGEN kit because it is most preferred kit, and you will get a good amount of RNA, and we can, if we understand from one type of kit, you can go with any other kind of kit where they have isolated RNA from the whole blood. So in previous lecture I have explained the basis. Now today we're going to learn the protocol, and the protocol, there are 5-6 steps. First is your RBC lysis, where you have to lyse the RBCs, complete removal of RBCs. Then leukocyte lysis, you have to lyse the leukocyte. Then you have to pass through the columns, then you have to wash the columns with buffers. Then you have to give a dry spin, which is very important to have a good the quality and quantity of RNA. And last one is elution. Depending upon your whatever concentration you want to have, you can increase or decrease the elution buffer. The kit says minimum of 30 microliter you can use for the elution buffers or to elute the RNA. So these 5-6 basic steps I have explained you. But before going to the utilizing these 6 steps, we have to learn each and every step in detail, which is very important. The first step is your RBC lysis. So for RBC lysis, what you have to do, you have to take 1 ml of whole blood. Then you have to add 5 ml. That means 5 times of your whole blood. Whatever amount you are using, 5 times you have to use your EL buffer, which is provided in the kit. Then you have to incubate. At 4 degree or ice for 10 to 15 minutes, right? And then in between you have to vortex. To ensure why we need to vortex? Because EL buffer is hypotonic to RBCs, and RBC cells, they will lyse in this buffer. But to ensure the complete lysis, you have to vortex. Vortex will ensure the proper mixing of all the cells, and it will also help in the RBC lysis. And now you have to spin down 400 G for 5 to 10 minutes at 4 degree. Now you have to be very careful. You have to see the palette. If your leukocyte palette is clear or transparent and your supernatant is red in color because of RBC lysis, so discard the supernatant and check the leukocyte palette. If your leukocyte palette is transparent, then there is no need to increase the incubation time. But if your leukocyte palette is not transparent, then you have to increase the time from 10-15 minutes to 20 minutes. If you give more incubation time, then means it will ensure the complete lysis of your RBCs. The leukocyte palette, point to be noted, the leukocyte palette should not have RBCs. So it should be transparent. Otherwise, the heme part will inhibit your PCRs or downstream applications. So now this is your palette. Now one thing you can do that once you have spin down, now you have leukocyte palette. What you will do? You will add 2 volumes of EL buffer. Again, you will add EL buffer to ensure the complete lysis of your RBCs. So, if you are taking 1 ml of blood, you have to add 2 ml. 2 ml of, in first step you have added 5 ml, now you have to add 2 ml. You have added 2 ml, now again you have to incubate for 5 minutes, and then you have to centrifuge or spin down 400 G for 5 to 10 minutes. Same step you have to repeat. So this is the first step, that is RBC lysis. Now we'll go to the second step quickly. The second step is leukocyte lysis. You have a palette, transparent palette of leukocytes. So these leukocytes should be lysed properly. Means we have to ensure that, because number of cells is if you are taking 1 ml of blood, that means it has 4,000 to 7,000 of leukocyte cells, and you want to have maximum amount of RNA from that group of population. So when from the leukocyte, you have to ensure the complete lysis of the leukocyte. So for that, they have taken a RLT buffer. This RLT buffer has chemicals which prevent the RNA degradation. That means, and it also inhibit the activity of RNases. Right? It will prevent the RNA degradation and it inhibits the activity of RNases. Along with this RLT buffer, you cannot use RLT buffer alone. You have to add some denaturing agent, beta-mercaptoethanol, BME, or DTT, diethiotheratol. So now, how much RLT buffer you have to add? If you are taking suppose 0.5 ml of whole blood, then you have to add 350 microliter of RLT buffer. And if you are using more than 0.5 to 1 0.5 ml. Then you have to use 600 microliter of RLT buffer. RLT buffer. So this is the table what I told you, that if you are using less than 0.5, then you have to use 350 microliter of RLT buffer. If you are using more than 0.5 to 1.5 ml, then you have to use 600 microliter of RLT buffer. But with this buffer, you have to add BME and or DTT, whichever is available in your lab, because that will denature denature the RNAs, and it will ensure RNA, even it denatures the RNA also, right? So your strands will open up, RNA molecule will open up, and it will help in the elution or binding enhance the binding of RNA molecule with the silica membrane. So now what you have to do, you have to vortex and then you have to spin down, right? Once you have spin down, so because there are several methods which can ensure the lysis, QIAGEN, they provide QIA shredder, they provide QIA shredder columns, which will ensure the complete lysis of your leukocytes, so that you at the end you will have good amount of RNA. So what you have to do, once you have added this BME or DTT containing RLT buffer, you have to vortex, spin down and transfer the supernatant Now what you have to do, transfer the supernatant to QIA shredder columns, right? QIA shredder columns, it will, as I told you, this is one of the method to lyse the cell other than homogenizers or rotor based methods. So once you have transferred to the QIA shredder, you have to centrifuge at maximum speed, that is 12,000 to 13,000 RPM. And now what you have to do, you have to, once you have centrifuge the homogenate, you have to preserve and discard the column. What you have to preserve now? Homogenate, because now all the leukocytes, they were lysed with the help of these QIA shredder columns. Now the next step is very important step, that is addition of alcohol. As we know that this is one part, and alcohol helps in the precipitation of the nucleic acids. So alcohol is added to the that homogenate or supernatant, you can say that. And how much you have to add? Either 350 to 600 microliter, right? Now give a vortex for 10 to 15 seconds, just to mix the ethanol with that homogenate, so that RNA can be precipitated, right? So now this is your second step. So to go with the third step, that is pass through the column. This mixture, that means homogenate plus alcohol, you have to pass through the columns, and the capacity of the column is 600 to 700 microliter. So transfer the amount or homogenate mixture to the column, to the spin column, right? अब आपको क्या करना है कि जो homogenate और alcohol का जो mix है, that you have to pass through the spin column. Once you have passed through the spin column, you have to spin down, that is 10,000 RPM for 15 to 30 seconds. Now what will happen? All the RNA molecule which are precipitated by the alcohol, it will bind to the column, that is silica, and once it is bound to the silica, now you have to discard the flow through. You have to discard the flow through. Remember, this is the important step. Once you have discarded the flow through, now this is your silica and this is your column, silica membrane. Your nucleic acids are attached to this. Some people, what they used to do, they add DNA also, DNAs, and this DNAs will lyse or will help in the degradation of the DNA, which is contaminating. To remove DNA treatment is given to remove the DNA which is present as a contaminant. Why DNA is present? Because this silica molecule is not specific, or the silica membrane is not specific for the DNA or particular RNA. It will bind to the nucleic acid, right? So it has DNA molecules also precipitated along with or bound to the membrane along with RNases. So some people, they give here DNase treatment, and the DNA digestion on the column people they used to do, but it's better to, you can give DNase treatment later also. Once you have RNA eluted, you can give DNase treatment later also. So this is your optional step. So you can go through the DNase treatment also. So what you will do now, once your homogenate with alcohol, that mix, is passed through the column, either you can go with the DNase treatment or either you can move ahead. So the next step is washing. The fourth step is your RNA molecules or nucleic acid molecules, they are bound to the membrane. Now what you have to do, fourth step is washing of the column. Now you have to wash the column. So for washing, you have to use RW1 buffer. You are adding RW1 buffer to the column, and then you have to spin down, same 10,000 RPM for 15 seconds, and then again you have to add 500 microliter, this is 700 microliter, 500 microliter of your RPE buffer to the column. This is again washing. Now 2 times we have washed. Now again you have to spin down 10,000 RPM for 15 seconds. Now you have washed your column. Once you have to wash, you have washed, again you have to wash with 500, you have to repeat the same step, 500 microliter of RPE buffer. Twice you have to do this, right? Then you have to spin down and 10,000 RPM for 15 seconds for 3 minutes. Why 3 minutes? Here is 15 seconds and here is 3 minutes. The difference in the speed, it is more than 10,000, that is 14,000 maximum speed you can do. Why this is like that? Speed is also increased and time is also increased, because this will ensure the complete removal of the alcohol which is present in the RPE buffer, so that if you increase the time of spin and the speed of the centrifuge, then it will remove the alcohol which is present in the along the walls or along the membrane, along the rim or whatever. So it will remove the alcohol from the spin column completely. That's why the time is increased. Here I want to tell you that the company people, they say that 14,000 RPM spin for 3 minutes is more than sufficient for the complete removal of alcohol. But if you want to have like a good quality of RNA, which is organic solvent free, so you can again spin down for 1 minute, which we explain as a dry spin. You can give one extra spin for 1 minute at 14,000 RPM, right? You can spin for 14,000 RPM for 1 minute. This is known as dry spin. So dry spin is very important to ensure the complete removal of the alcohol. Now your column, your membrane, your your collection vials, all are free from the alcohol. Now dry spin is very important. Sometimes company people say 3 minutes, 13,000 is enough after your RPE buffer, but I preferred one extra dry spin at higher speed for 1 minute, which ensure the complete removal of your alcohol. Now what you will do, fifth is elution. What you will do, you will change change the collection vial, with 1.5 ml of MCT, microcentrifuge tube, and place the column over it. And once you have placed the column is also covered with lid. So once the column is placed, you can add exactly to the center of the membrane the elution buffer. One very important point to be noted here, that add elution buffer to the center of the column, not along the wall of the column. Because why? Because sometimes what happens, the elution buffer, because the amount is very less, so elution buffer, it will remain as a bubble here, or it will not go completely to the base of the silica membrane. So to avoid that incomplete elution, or to avoid that less amount of elution buffer to be in contact with the RNA, you have to add your whole either 30 microliter or 60 microliter, whatever elution buffer you are using, to the center of the membrane. And don't touch the walls here and here and go directly to the center, and change the tip every time. Change the tips every time while you are adding elution buffer, because otherwise it will contaminate the other samples also. So now you have added 30 microliter to 60 microliter your RNase free or nuclease free water. You can say nuclease free water exactly to the center of the column. Now wait for 1 to 2 minutes, because give sufficient time, 1 to 2 minutes, for the elution of for the elution of maximum amount of RNA from the silica membrane to the elution buffer. Now last step is you have to centrifuge approximately 10,000 RPM for 1 minute. Now you have eluted, you have 30 to 50 microliter of eluted RNA from whole blood. So these are the basic steps where we can isolate the RNA from kits like Qiagen, which are provided by Qiagen, and this is the basic. So in next lecture we'll learn what to do with this RNA. How to preserve this RNA, and how we can prevent the contamination or we can degradation of RNA from the RNases. There are some steps and precautions which we can take. And one thing, you have heard about DEPC water, and DPEC water is very it is used in many labs. How to prepare that is very important. So we'll learn that. And DEPC water is important to decontaminate your apparatuses, also to clean your apparatuses finally, and it is also important to dissolve the RNA also. So that we'll learn in the next lecture. So this is with the elution of RNA from the kit based methods. And if you have any queries, any doubt, you can ask me in the comment section. Thank you.